Good afternoon, Year 13. So we're going to have the first lesson in the nervous system topic here. Obviously we did the other lesson as a live lesson, and this one was a live lesson too, but you've got this recording for now. So, first things first, from GCSE I need to make sure everyone understands these points here. They're important, it's something you need to know before you can get started. So pause me, do these 5 questions, just the answers, and then we'll carry on. Okay, hopefully you pause me. Hopefully, otherwise you won't have had very long. The different methods of messaging employed by the systems. Okay, so the nervous system is using electrical impulses that are along nerves to transport information around the body. The endocrine system is the hormone system, so it's using chemical hormones around the blood. Which is faster by an enormously long way. The nervous system is much faster. How are specific cells targeted by each? Well, the nervous system targets cells by directly interfacing nerves with those cells. So the nerves have to directly attach onto cells or onto cells that are attached onto nerves. So broadly, the nerve physically attaches itself onto a cell and electrically stimulates it. Endocrine system works by hormones attaching to receptors on the outside of cells. Which last longer and which can be permanent? I realise are very similar questions. Hormones. Hormones last an enormous amount longer and can have permanent causes, permanent effects, which the nervous system just doesn't have. So having understood that, and particularly number 3, how we're actually targeting the cells, then we're going to move on to some more A-level stuff. And basically what we want to know from today is how nerves maintain their steady resting state and why that's important. I'm going to start off by talking about the structure of a neuron. So neurons are basically nerve cells that specialize for the transmission of impulses to other neurons and cells. So basically there are 3 types, and you learned about these at GCSE. There are sensory neurons, relay neurons, and motor neurons. Now sensory neurons, sometimes called afferent neurons, let's just lead with the word sensory, they transmit impulses from receptors to the central nervous system. So we're talking about a light being shot in my eye, let's say, a light receptor at the back of my eye, then activates a sensory neuron. A relay neuron are the neurons that are found inside the nervous system, inside of the central nervous system, inside your brain, your spinal cord. They coordinate that information, pass it from sensory to motor neuron. And then a motor neuron, which is sometimes called an efferent neuron, transmits impulses away from the central nervous system to a muscle or a gland. Now all of that should be familiar to you from GCSE. So again, kind of repeating ourselves. This is going to be less familiar, I think. So to start with, we've got these things called dendrites. Now dendrites are where information is going to come into the neuron. So electrical impulses are going to start at the dendrite. The dendrites are therefore either going to be attached to receptors, if you're a sensory neuron, or in the case of a motor neuron like this one, they're going to be attached onto other neurons, usually relay neurons in this case. Now those dendrites lead straight into the cell body. Now the cell body is kind of your standard cell bit. It's where your nucleus is, your rough endoplasmic reticulum, and all of that stuff is going to be in your cell body. Now the cell body does all of the stuff that the cell needs to keep it alive. It doesn't do any transmitting of impulses. Coming out of the cell body is usually one axon. Now an axon is the way that the information is then going to be transported onwards. So we get an electrical impulse from a relay neuron, let's say, into our motor neuron. That motor neuron will then be depolarized, be activated, and will send that impulse down the axon. In this case, it will interface with some muscle cells down the bottom. Now, around these axons can be things called myelin sheaths. Now, myelin sheaths are basically made of fatty cells called Schwann cells, and we covered that in the other lesson. But broadly, we're looking at this nerve cell being covered by this myelin sheath, and it's going to make the transmission of the impulse faster. And then down at the bottom we have these things called axon terminals, and that's going to be attached directly onto, in this case, a muscle via something called a synaptic knob. Everyone's favourite term in biology, but this synaptic knob is just where this synapse is going to happen. Now we're going to cover each part of the nerve in a huge amount of detail. Now the lesson we just did on live covered the axon basically. We're going to talk about the dendrites in a separate lesson. We'll talk about the synaptic knobs in the third lesson. And always the impulse runs from dendrite through cell body, through axon and into the synapse. Never the other way. So, one thing I need you to understand is that neurons and nerves are not the same thing. Nerves are made of nerve fibres, and nerve fibres contain the axons of neurons. So basically, a nerve is just a bundle of a lot of neurons. Thousands, hundreds of thousands of neurons. Now, around this entire thing is connective tissue. Basically just holds it together, protects it, so it's not getting damaged easily. Now, these things can be either just entirely motor or entirely sensory, or they can be a bit of both. Now, it's worth noting you don't need to understand that particularly, except for the idea of what the difference between a nerve and a neuron is. At no point should you write a nerve when you mean neuron. A nerve is a bundle of neurons. And if we're looking at a microscope image of a human nerve, you can see every one of these white circles with black around it. The white is the axon, as if you've kind of sliced it and then you're looking into it. And the black is the myelin around the outside of the neuron. But this whole thing is one nerve with lots and lots and lots of neurons. Right, 3 questions there. Have a go at them. Pause me. So, the name of the extensions that stretch out of the cell body, that's an axon, or you could say dendrite. Now, they both stretch out. The axon is the one going to, with the electron impulse, going to travel from the cell body to other neurons, to the effector. The dendrite is going to be where all the impulses come from. The name given to the covering that surrounds some individual neurons is a myelin sheath. True or false, each nerve is made of a single neuron. That's very false. They're made of many neurons. Fine. Now, this is ridiculously important, this bit. We understand this. Now, it's true for pretty much every cell, but it's particularly important for neurons that there is a relatively positive charge outside the axon and a relatively negative charge inside the axon. Now, this is caused by a lot of things, but basically what keeps it like this is the fact that this membrane is made of fat, phospholipid. Phospholipid does not transmit electricity well, and so we can't equalise that. The positive charge is, if you like, stuck outside, and the negative charge is stuck inside. And this is called a membrane potential. And when we talk about the membrane potential, and we're going to say things like minus 70 millivolts, plus 40 millivolts, I'm talking the inside relative to the outside. So when I say that the current, looking at that, the current membrane potential is minus 70 millivolts, I mean that overall, compared to the outside, the inside is 70 millivolts less. Fine. Now, I'm not a physicist, I don't claim to be, I just need to know that number, minus 70 millivolts. It's called the resting potential. In any case, throughout this, I should have mentioned you should be pausing me to take any notes you think are necessary. Fine. So, this is a very important graph. Now, when a neuron is activated, it causes something called an action potential. Now, an action potential is basically how a neuron is activated. It causes this electrical impulse. And what it looks like is this. It's a temporary reversal of the membrane potential, i.e., usually we said the membrane is the inside is negative relative to the outside. Suddenly, the inside becomes positive relative to the outside, so the outside becomes negative. That's called depolarization. So we start off at minus 70-ish, and we're going to suddenly depolarize, and we're going to become more positive inside than outside, before eventually repolarizing, and occasionally hyperpolarizing. Basically going back to where we were before. Sometimes we go too far downwards and we have to bring it back to normal again. Now that is what we're going to look at a lot later on, and you will have covered it in our live lessons, so I'm not going to dwell on it too much. But we're talking about changes in the positivity or negativity of the inside relative to the outside. Fine. Have a look at those notes, decide what you want to take down. So, in order to understand this next bit, we have to know the answer to these three questions. The first two I'd expect you to know off by heart. The third one is more confusing to you if you're not a chemist. I will talk to you about that. What do you know already? So, what is the chemical charge of a potassium ion? Hopefully you're thinking in your head immediately, K+, positive, just one positive charge. Now, I don't even need to know that it's one positive, just plus. I only need to know that it is positive in general. The potassium is positive, and that's fine. How about sodium ion? Well, again, I'm hoping you're thinking alkali metal, GCSE, Na+, and so it's positive again. And then a protein, I don't necessarily expect you to know this, but proteins generally have negative charges overall, and that's just something we need to know. Now, what we're going to talk about now is how we keep the inside of the neuron negative relative to the outside. And we do that by controlling the location and concentration of the different ions, potassium and sodium particularly. And basically, where there's more of those two, they'll have a positive charge. So if we know that the outside of the membrane is positive, we probably know there's more potassium and sodium overall outside the cell than inside the cell. So, we're looking at something that looks a bit like this. Now, broadly, you can only let sodium and potassium across where we have carrier proteins or channel proteins. And broadly, there are two types of channel proteins we're going to talk about. And remember, channel proteins are allowed to facilitate diffusion. We're talking about voltage-gated channels, and basically they open up at when you have a certain voltage, i.e. when you get depolarized a certain amount. And those are found for both sodium and potassium. And then we have leak channels. Now, leak channels are protein channels that remain permanently open, i.e. you can't close them, and they only exist for potassium. So there are leak channels for potassium, but there are no leak channels for sodium ions. Fine. Now, once you've understood that kind of structure, we can start talking about how we're maintaining this resting potential. And we're going to do it by keeping these leak channels open. Now, broadly, this is controlled by sodium-potassium pumps. Now, these are active transport proteins that are working continually to pump potassium into the cell, potassium ions, I should be careful, and sodium ions outside of the cell. Now, you might think, well, that's completely positive. That's completely unnecessary, excuse me, because they're both positive. I'm just pumping one positive charge one way and one positive charge another way. But we now need to think about, if I'm pumping the potassium ions in, I'll have a ridiculously high concentration of potassium ions inside. If I'm pumping the sodium ions out, I'll have a high concentration of sodium ions outside. So what's going to happen? Well, the potassium ions can escape. They have these leak channels. They're open all the time. So as I pump potassium ions in and increase their concentration, they will move out again by diffusion. The sodium ions, once I've pumped them out, they have no open channels when the membrane is at rest. So they can't get back in. They can't get back in, and so you end up with all of the sodium ions pretty much being pumped out and getting stuck there, and all the potassium ions being pumped in, but then diffusing back out again until you have about the same concentration on both sides of the membrane. So then I've got about half the potassium outside and all the sodium outside. So there's a load of positive outside compared to inside, and I have these negative proteins inside. And so overall the inside becomes negative. So this is basically what we've just said. In fact, it's exactly what we've just said. Feel free to copy down any notes there. Fine. So we end up with something like this. And overall, we have a lot less positive charge inside, and we have a lot of negatively charged proteins and some other ions we're not going to talk about, and so the inside becomes negative. We've talked about that. That's fine. Now this only really stops when you get down to this negative 70 mark. And that is because, basically, it's so negative inside that actually the positive charges on the potassium begin to get attracted in. So now the potassium wants to move out by diffusion, but it wants to move in because it's attracted to the negative charge. And eventually you end up with an equilibrium where you get roughly net movement of nothing, where nothing's moving out, but overall nothing's moving out and nothing's moving in. Because some of it's going to move in because of the electrochemical, some of it's going to move out because of diffusion. It's going to balance itself out. And this is how we rest. We keep pumping any sodium ions that are created inside the cell via chemical reactions and released out. We keep pumping potassium in, and potassium keeps moving out, and so we equilibrium, we rest at minus 70 millivolts. Now, 3 questions there. Again, pause me. So, the 2 types of protein channels are leak and voltage gated. Potassium ions can go through the leak channels, but only sodium ions can go through the voltage gated. Sodium ions, can only go through voltage gated. Potassium ions can also go through voltage gated. What stops the membrane resting potential becoming more and more negative? Well, overall there's an electrochemical gradient, so the inside becoming negative attracts the potassium back inside again. And what is an action potential? It's a temporary reversal of that charge from negative inside to positive inside. Right, now that's all we're going to do for now, and we came to this next bit of the lesson in our live lesson. Hopefully that's been helpful for you. If you have any questions about any of that, please feel free to contact me on